Okay, I think we can get started. I want to welcome everybody to today's OCB webinar. My name is Heather Benway. I manage the OCB project office, where I'm joined by two other staff members, May Mahegan, who manages OCB communications, and Mary Zawoski, who manages administrative and logistical operations. I'd like to acknowledge the sustained support of the NSF Chemical and Biological Oceanography and NASA Ocean Biology and Biogeochemistry programs for making the OCB program possible, and also the Woods Hole Oceanographic Institution for hosting the OCB project office. Today, we're also joined by OCB SSC member Emily Osborne from NOAA AOML, who is serving as our webinar co-host. To give you a sense of the scientific breadth of OCB, these green icons depict our overarching science areas, and you can find more specific research questions of community interest on our website on the About Us tab. We invite you to engage with the OCB network in the many ways listed here. Please visit our website, sign up for our biweekly e-newsletter, and/or follow us on social media. You can also reach us by email at the address listed at the bottom. Just to note that there is a hashtag for the webinar series that you can feel free to use during or after this webinar. Just to share a few upcoming webinar teasers, OCB is partnering with the North American Carbon Program on a joint mini-series of webinars focused on extreme events in the carbon cycle. These webinars are intentionally scheduled to precede the virtual 7th NACP Open Science meeting, which is scheduled for Friday afternoons in March. The speaker lineup looks amazing and registration is free. Check out the info on the bottom of this slide. These webinars, these joint webinars between our program will actually set the stage for a breakout session during this meeting on March 19th that's going to focus on extreme events. And we're hoping to use these talks as scientific fodder for discussion. We'll be bringing interested scientists and stakeholders together to plan future research, develop new collaborations, and interact with each other in planning and discussion. So I'm hoping that, I know I'll be joining, and I hope many of you will join as well. Here's another OCB webinar that's coming up after our special mini-series that I referenced before. This is will be the next OCB webinar in late March, focused on ocean iron cycling. So OCB webinars typically have a topical theme with 2 speakers per webinar. If you want to give a talk, please submit an abstract. The link is on the slide, and we'll also put it in the chat. And if you have ideas for thematic webinars, our format's really flexible, so please do get in touch. To broaden access, all webinars are recorded and they get posted on our YouTube channel within a few days. We share all information about our webinars on our website, our e-newsletter, and on Twitter. So again, connect with us in all of those ways and you will always be informed. Here's a short screenshot of the attendee Webex panel with key features highlighted. I just want to point out a couple of things. Please note that all attendees are muted with video off just to reduce background noise and distraction. On the bottom right-hand side is the chat, which is this little messaging speech bubble icon. And also, if you click on this ... right next to the chat, you can find the Q&A function, which you can use to ask questions. If you're entering questions into the chat, please select everyone or host and panelists as the recipient of your questions. Otherwise, most of the panel won't see them, only the host will. Both Emily and I will be monitoring and moderating the Q&A section. We'll have a short Q&A after each set of talks. So the theme of today's webinar is Regional and Global Perspectives on Ocean Deoxygenation. Today's speakers are Mathilde Jutras from McGill University in Canada and Marina Levy from the Ocean and Climate Laboratory at the Sorbonne in Paris. After today's webinar, we're going to have a short little informal chat and Q&A with the speakers. Early career attendees are especially welcome. All are welcome, but in many cases, we like to make sure that people are comfortable. We're having this as an informal event so that people can ask questions and and not feel intimidated in any way. This will start at about 12:15, so you get a 15-minute bio break before rejoining, and we'll put the link in the chat for joining in this informal meeting. Now I will hand it off to Emily to introduce our first speaker, Mathilde.
Thank you, Heather. It's my pleasure to welcome all of our attendees today and our presenters as well. I'd like to introduce our first speaker, Mathilde Jutras, who is a PhD candidate in earth scientists at McGill University in Canada. Her research focuses on understanding the physical and biogeochemical processes leading to the deoxygenation of the St. Lawrence Estuary, which we'll be hearing about from her today. This research topic has led Mathilde to her interest in ocean circulation and climate in the Western North Atlantic Ocean. Mathilde holds a master's degree in atmospheric and oceanic sciences, also from McGill University. And following her master's work, she worked on urban climate governance and adaptation and is strongly engaged towards sustainability questions. Speaking of which, she launched the Responsible Research Initiative to raise awareness within the scientific community to the environmental impact of their academic travel practices. While COVID-19 has slowed down travel and her awareness campaign, she hopes the pandemic will be an occasion to rethink academic travel and make virtual conferences and exchanges more common. Since 2020, Mathilde has also been a member of the Intersectoral Student Committee that advises Quebec's Scientists-in-Chief. So without further ado, Mathilde, please take it away.
Hi, thank you very much for the presentation. And so I'll share my slides. Hopefully everyone can see it now. So thank you. So yes, I will present this talk today titled Changes Through Time in the Causes of Oxygen Decline at the St. Lawrence Estuary from Biogeochemistry to Circulation. So you will see what this is all about. My research interests are on the causes of deoxygenation in that area, but you'll see that this leads me to also get interested into what is happening more broadly in the Western North Atlantic Ocean. So I'm co-supervised for my PhD by Carolina Dufour at McGill, and Alfonso Mucci, who's a chemist also at McGill. So, and this presentation is mostly based on results that we recently published in JGR Oceans. If you want to see more details, you can go have a look at the paper. So just to start off with a little context, most of you know that the ocean has been losing oxygen over the last decades, and you've probably seen this figure before, where you see the oxygen loss in the top and bottom ocean, in the top and bottom panels. In total, the ocean has % of their oxygen content since 1960, and but as you can see, there are some very large regional variations in these changes. And coastal regions also experience deoxygenation. So on this map, all the red dots show the documented hypoxic areas. So these are areas where the oxygen is low enough so that the conditions will be harmful to most marine species. And the blue zones show coastal areas where you have a large zone of low oxygen concentrations known as oxygen minimum zones that the next speaker, Marina Levy, will talk more about. In coastal areas, one of the processes leading to hypoxia is eutrophication. So for those who are not familiar with it, it's the process by which if you have an excess of nutrients that comes into the system, it will lead to algae blooms at the surface. And when these organisms die, they sink to the bottom of the ocean or of the water, and they get decomposed by bacteria that do respiration to decompose them, and so they use oxygen. And this is why the oxygen concentrations in the bottom of the waters will get depleted due to this extra amount of nutrients. And without going into all the details, there are many processes that lead to deoxygenation in the open ocean and in coastal environments. Like, for example, the increase in temperature will lead to a drop in oxygen solubility. Changes in stratification due to the warmer waters will also affect reoxygenation of the deep waters. There are changes in circulation, etc. And in some cases, like in the St. Lawrence Estuary, you have both coastal and open ocean processes acting together to reduce the oxygen concentrations. Okay, so where is this estuary? Here you have North America, and The enclosed sea that you see here is what we call the St. Lawrence Gulf and Estuary. And we will focus more on this part here, which is, well, basically like the less broad area here is what we call the St. Lawrence Estuary. If we zoom a bit more, so this is our region, and where you have the pink star is the end of the estuary, and the waters become much shallower as they're forced to upwell, and this brings a lot of nutrients and lead to very active waters that attract a lot of whales, where people go whale watching, including the iconic beluga that you see on the picture here. And this is also the location where the deep waters of the system are hypoxic. As you can see on the figure, there was a strong decline in the oxygen concentrations since the 1930s when Clarence first took measurements up until now. Okay, so this is what the system looks like along the estuary. So here we have Quebec City, and here we would have the Atlantic Ocean. So the estuary is characterized by a deep channel of about 350 meters deep, and the waters upwell at the end, as I mentioned. So typical of an estuary, you have the deep waters moving landward and the surface waters moving seaward. The system has 3 layers because in the winter you have cold, like a winter, you have convection that will lead to the creation of this cold intermediate layer that will remain throughout the year. And the waters take between 4 and 7 years to travel from the Atlantic to the head of the system, and this is a very long time. So during that time, the waters have a lot of time to get depleted in oxygen. And this is what we see on the figure here on the map. The color shows the oxygen concentration at the depth of the oxygen minimum. So you can see the progressive depletion as the waters move landward. So it means that the oxygen concentration will be naturally low at the head of the estuary, but the drop that you saw in the previous slide is attributed to 2 types of causes. So first we have the more local biogeochemical causes that are caused by, for example, the increase in nutrient export into the St. Lawrence River, which is quite high because the watershed contains a lot of areas with industrial agriculture that will use a lot of fertilizer, and also a very large number of big cities that release their sewage water into the system. So we have a strong load of nutrients. We also have a local increase in deoxygenation due to climate, we could say, because it's due to the fact that the waters are getting warmer and hence the biological activity is stronger. Second, we have the external climate-related causes, because the initial oxygen concentrations before the water start their transit here is set by the ratio of Gulf Stream and Labrador current water that enter the system. And and the Labrador current water are colder, which means they have a higher oxygen content, and the Gulf Stream waters are warmer, which means they have a lower oxygen content, and therefore the ratio will set the initial oxygen concentrations. So previously, we've seen that there is an increase in the amount of Labrador water coming in, and this was attributed to a slowdown of the AMOC in a paper. I think, yeah, here I have the figure. So in a paper by Clarence in 2018, they could see in a model, if here you have a pre-industrial run, and you can see that there are a lot of oxygenated Labrador waters that can reach near the entrance of the system, but not in the 1% CO2 run. And they attributed that to a retreat of the AMOC that was driven by climate change. All right. So we know a bit what is happening, but we don't know exactly what is happening, and in particular not through time, if the causes are changing and are they related to climate change, etc. So this is what we will investigate here. So we use a method that is a bit different to what is typically used, because typically to look at the mixing of water masses, we will use TS diagrams. So on the left here is the analysis that was made in the St. Lawrence Estuary that I mentioned before, where they saw the increase of Labrador current waters. So we have two sets of points that represent here the circles are the Labrador waters and the triangles are the Gulf Stream waters. And we take measurements in the estuary and we put them in the diagram. So these are the squares here. And the distance from the two lines will give the fraction of each water mass. However, this analysis assumes that the mixing is isopycnal because we draw a straight line here. And this might not be the case that we have isopycnal mixing where we have rough topography. And here this is just a generic plot from Paulsen 1997 in the Brazil basin. And you can see that the diffusivity is much higher over rough topography. And this would probably be the case near coasts where we have shelves and channels and things like that. So the TS diagram analysis might not be appropriate near shelves like in our area of study. Therefore, to study the causes of deoxygenation, we use a method called the extended multi-parameter analysis that reconstruct the fraction of water masses contributing to a measurement. So I will present the method quickly just because it's very easy to use and I think it could be interesting for many people, but I'll try to keep it simple. So basically the EUMP is just a fancy name to say that we solve the linear mixing set of equation for a number of parental water masses described with a number of variables. So we say that the fraction of water mass 1 times, let's say, temperature of water mass 1 plus same thing for water mass 2 will be equal to our observation. We use that are conservative upon mixing, like temperature, salinity. But in the extended version, we can also use variables that covary as long as they covary according to a fixed ratio. And this is the case for oxygen and nutrients in respiration and photosynthesis. So this is quite useful. So we get two outputs from the calculations. The first one is the fraction of each water mass, so the x's here. And the second one is because of this addition of the semi-conservative variables, we also get an estimate of nutrient or oxygen consumption compared to the unaltered parent water masses. So both of these outputs will be of great use to reconstruct the oxygen budget. So just to finish off, the EUMP has a couple of advantages over the simple TS analysis, apart from the fact that we can get that estimate of oxygen consumption. So we can use a large number of parental water masses. We are not restrained to 2 or 3. And we assume nothing about the mixing, as I mentioned. Okay, so in our system, to run the analysis, we need to identify the parental water masses that contribute to our system. In our case, we will ignore the surface waters, and we consider 3 parent water masses that feed the system. So the Labrador current waters, as I mentioned, that we define as located here. Then we have the Gulf Stream waters, and we have also a local water mass in the system just for the intermediate layers to close the budget. We use 7 variables, and I used all the observations I could find that would have these 7 variables. So this is some data in the 1970s, and then a continuous time series from 1995 to 2018. Okay, so a first result that we get from this analysis is the distribution of the parent water masses in the system. So here you see cross sections along the channel, so along the pink line here, of the fraction of the different water masses. So here is the density, and here is the Atlantic with depth. And the first plot shows red is where you have a lot of water from the CIL, and blue is where you have none. So we see that most of the water in the intermediate depths fits with that. So this is good. And you see that the bottom waters are composed of a mix of Gulf Stream and Labrador waters, with the Gulf Stream waters lying slightly above the Labrador current waters, which is interesting. And they mix progressively as they move inward. And so this is for 1 year, for 2017, but we can do the same thing for all the years. And yeah, okay, and sorry, I'll come back to that after. Yeah, so for all different years, and if we want to look at the contribution that enters the system, we can concentrate on the red box here, so at the entrance of the system, and see what is the fraction through time. So this time series is shown here. So what we see is that first, the Labrador current water contribution decreased from the 70s and the Gulf Stream contribution increased from the 70s. We see that there is quite a low interannual variability, and it looks like there is no, well, there is no significant trend between the early 2000s and 2008. And in 2008, there is a strong decline that begins and that lasts until the end of the time series. Sorry, I forgot a slide here. So I just want to come back here and present these results just to compare what we would have obtained if we would have used the TS diagram analysis. So this is what the analysis would have looked like, similar to what I showed before. And we would obtain the stratification shown in the middle here. So with Gulf Stream waters in the bottom and Labrador current waters more like on the top. And this I showed before for the EOMP. So you can see that the stratification and the distribution of the water masses is different between the two. And this suggests that there was probably some diapycnal mixing that occurred when the two water masses met near the shelf and then entered into the channel. And this really suggests that we should use this method instead of the TS diagram. And it can also be used to study if there is a presence of diapycnal mixing or not. Okay, so coming back to this here. So from this, we can see the change in oxygen that is due to the change in the ratio at the entrance of the channel. But if you remember, the EOMP also gives us an estimate of oxygen consumption. And from that, we can get a time series of oxygen utilization between the parent water mass formation location all the way to the entrance of the channel in blue here, and also along the Laurentian channel, oxygen utilization between like the entrance of the channel and the end of the channel in black. So what we find is that the oxygen utilization increased in the estuary until 2000, and then it stabilized. And this fits, the bottom plot shows input of nutrients with time in blue and fertilizer sale in black, just to compare, for two nutrients, nitrogen and phosphate. In the St. Lawrence, it was shown that nitrate is the limiting nutrient contributing to eutrophication, so we focus on that curve here. And we see that the increase, here we don't have any data in between, but the increase fits with the fact that nutrients stabilize after. So we can kind of say probably what was causing deoxygenation in that time period was the excess of nutrients. And we also find an increase in oxygen utilization in the North Atlantic, but I have no explanation for that for now. So we can combine all of this, so the changes in the Gulf Stream to Labrador ratio and biogeochemical changes, to get a budget of the different deoxygenation. So the pink bar, oh yeah, and we're going to focus on two periods. If you remember, in 2008, there was like a shift in the time series. So we're going to focus on the 1970 to 2000 period, and then 2008 to 2018. So this pink bar shows the total measured deoxygenation. And on the right, we will pile up the different causes. So first we have green and orange are the change in oxygen consumption in the channel, so eutrophication. And the orange part is due to the increase in temperature. Then we have the change in oxygen or in biogeochemistry in the North Atlantic. And the blue is the deoxygenation due to the change in the Labrador to Gulf Stream contribution at the entrance of the channel. So what we find is that the causes of deoxygenation are not constant with time. And during the first period, it's really the biogeochemical changes that dominate the total deoxygenation. And in the second period, it's the circulation changes in the North Atlantic that dominate. So I want to finish by asking the question of what causes this change in 2008 or in general in the contribution from the Labrador current to the system. So to do so, we looked at a number of indexes that characterize and control oceanic circulation in that region. First, what you see here is that we correlated the Labrador current contribution in blue, the same time series that we saw before, with the Labrador transport index over the Scotian shelf, which measures the amount of water that, like the transport in the zone here. So it's an extension of the Labrador current. And you see that the correlation is quite strong. In contrast, we find no correlation with an index of the Gulf Stream volume transport or with the Gulf Stream position, like for example, how far north it is or how close the front is to the entrance system. Therefore, it seems that the Labrador current and what is happening in high latitudes is what is controlling the properties of waters on the continental slope and in the St. Lawrence, and not the Gulf Stream. But this is something that we will explore more because this just opens up so many more questions. So to conclude, this figure shows the different contribution to deoxygenation along the transit of the waters. And as I mentioned, the causes of deoxygenation vary with time. In the first period, it is dominated by local biogeochemical changes. And the second period is dominated more by external and climate-related causes that are related to the Labrador current, apparently. So this local study shows how coastal environments can be subject to a complex interplay of coastal and open sea deoxygenation stressors, how they can add up together to worsen the deoxygenation or compensate each other. And this means that when we study deoxygenation in the open sea, we can also think of the additional impact it can have in some coastal regions where it's already on the edge of being hypoxic in terms of conditions. Thank you. So if you're interested, I invite you to have a look at the paper. I have the reference here, or to contact me for comments or questions or anything, and I'll be happy to take questions now.
Wonderful. Thank you so much, Mathilde. And we would be happy to take questions. So folks, you can enter your questions in the chat or in the Q&A. We'll give folks a minute to type. All right. All right, we have a question from Greg Cutter. Excellent talk. Could the lesser amount of Labrador water be due to freshening and buoyancy?
Mm-hmm. It's something we looked at. I didn't show the plot, but we looked at the properties of the waters on the shelf and on the slope in the Labrador region to see if there was a change in salinity or temperature. There was nothing significant and no trend during that period. So we assumed that the properties of the waters didn't change. So it wouldn't be due to that. But yeah, that's definitely something important to consider.
All right, next question is from Shiras Yipian. So sorry if I said your name wrong. Does this system experience anoxia or does it remain hypoxic?
Yeah, so it remains hypoxic for now. Hopefully it will stay like that. But yeah, it's a particular system because I feel like a lot of estuaries and coastal environments are shallow and will experience hypoxia that lasts for a couple of months or like seasonal. But in that case, it's so deep, the channel is like 350 meters deep, even at the end, that we have, these waters are completely isolated from the atmosphere. So yeah, it's only hypoxic. Sorry, I deviated a bit from the question, but.
All right, our next question is from Mariona Clarette. She says, wonderful talk, Mathilde. I have a question regarding the EOMP analysis. Do you need to know the temperature and salinity and member values of the parent waters to perform it?
Mm-hmm, exactly, yes. And of all the variables that you use, and since we use 7 variables, we need to have values for all the variables. So to do that, we use the climatology in the ocean, and on the map that I showed, the regions are like the regions that we use to get these values in TS diagram or S oxygen diagram. Yeah. And this is also why I mentioned that we consider they are constant. You could consider that they vary with time also, but you need to define your current water mass system.
I think you answered the follow-up question of how they're set, but there is kind of a more specific aspect of doing this for your cold intermediate waters specifically. So do your climatologies cover those?
So for the cold intermediate layer, we use the data that we have in the Gulf actually. So it's not from climatologies, but yeah, all the data that we had over the years.
There was a follow-up comment also from Greg, who was your first question, and he mentions the Gulf Stream is also weakening, so that's an added complication.
Yes, well, I mean, that's why we were surprised at the end to see that there was no correlation with the strength of the Gulf Stream. Because at first, that's why we would have thought that this weakening of the Gulf Stream would be what is causing this change. But it really seems like it's the Labrador Current, like the force of the Labrador Current that is driving the change, and not the force of the Gulf Stream.
All right, we've got another one. Does the EOMP assume that oxygen respiration rates are the same in both water masses?
So the ratio is the same in all cases, like we could say with time, but then it will give a different amount of. Oh, okay, I see what you mean. No, it can. Yeah, I'd have to think about that. I'm not sure. I don't want to say something that's not true.
Heather, do you have any questions queued up?
I don't see anything in my chat window.
Okay.
So with that, why don't we move on to our next speaker?
All right. Well, thank you so much, Mathilde. Wonderful, wonderful talk. So I'd like to go ahead and introduce our second speaker now. Marina Levy is a French National Center for Scientific Research, or CNRS, research scientist at the Ocean and Climate Laboratory at Sorbonne University in Paris, France. She received her PhD from Sorbonne University and did her postdoctoral fellowship at Lamont Doherty Earth Observatory. Her research interests include the interactions between ocean physics, biogeochemistry, and marine ecosystems. She is recognized for her long-ranging work on interactions between turbulent ocean currents and marine biology, with an ultimate goal of improving model projections of ocean stressors in response to climate change. She was president of the Ocean Science Committee of the French Space Agency CNS from 2014 to 2019, and since 2016, she is deputy director of the Ocean Climate and Resources Department of the French Research Institute for Sustainable Development, IRD. She is also associate editor of the American Geophysical Union journal, Global Biogeochemical Cycles. So join me in welcoming Marina.
Thank you, Emily. Hi, everyone. It's really a pleasure to be here with you today. I see all your names, and I see many friends and colleagues, and I wish I could see you in real. But still, it's great to know that you're here. Let me start by sharing my screen. Yes, and the pointer, laser pointer. Yes, here we are. So Ether asked me to give a global perspective on ocean deoxygenation today. And I wanted to maybe stress the fact that not all sites in the ocean are equally vulnerable to a loss of oxygen. And this is why I wanted to focus my talk on oxygen minimum zones, which are large areas, large bodies of water at mid-depths in the ocean, which are located over major upwelling systems, which are already naturally deprived of oxygen. Most of them are hypoxic, inhospitable to many marine biota, and in the core of some of them, there is even anoxic situations, which leads to denitrification with important climate feedback. If global deoxygenation is associated with an expansion of these oxygen minimum zones, which is not obvious, this would have disproportionate important consequences on the shrinking of marine habitats or the amplification of denitrification. Before digging in more in these oxygen minimum zones, I will just share 2 slides on global deoxygenation to maybe repeat some of the things that Mathilde has already said. One of the, among other tracers to the oceans, among other consequences that climate change have on the ocean, ocean deoxygenation stands out because it's something that really has already been observed. And as Mathilde said, we have observed a decrease of the oxygen content of the ocean of nearly 2% over the last 50 years. This plot shows the time evolution of the total oxygen content in the ocean taken from the IPCC Special Report on the Ocean and Cryosphere. And what our model projections are projected for the coming century is that this trend is projected to continue at a fairly similar rate under the business as usual scenario. There is a very large body of literature that explains why we have this deoxygenation, and I think it's fairly well understood that approximately half of the deoxygenation is due to a solubility effect, simply the fact that warmer ocean temperature can hold less oxygen. Here I'm showing one of these studies led by my students, Damien Coupieu-Spel, and which is based on an Earth system model IPSL under a business as usual scenario, where he's been able to disentangle the different causes for this total deoxygenation over the 21st century in this model. So in agreement with previous study, he's finding that half of it is due to a solubility effect, and the other half is due to a change in apparent oxygen utilization, or AOU, which is really the fact that the utilization of oxygen along the penetration, the ventilation of oxygen when it enters the ocean. And using a method based on the fluxes of oxygen across the base of the mixed layer, Damien has been able to show that the decrease is mostly due to a decreased rate of ventilation, which is only partly balanced by a decreased rate of oxygen utilization by respiration. But in fact, this global analysis doesn't really hold for oxygen minimum zones. If we put side by side a map of these oxygen minimum zones to this map, which shows here the trend in the loss of oxygen over the 21st century. This is an average over many SEMI5 model projections under the high emission scenario. You see these large areas of deoxygenation, mostly in northern latitudes in red, which do not coincide with these areas of OMZ, which are mostly in the tropics, and where there are large uncertainties in the model projections, which is shown here by the gray shading. Laurent Bopp has partly explained where this uncertainty comes from. If you separate between the change in oxygen saturation and AOU, you can see that in northern latitude, the two effects tend to add up. You have red on the two sides, but in the tropics they tend to oppose each other over OMZ. In fact, the effect of solubility is rather weak over the tropics, and it's mostly the effect of AOU which seems to lead the signal, and this signal seems to suggest that there is a decrease of this AOU, which could be due either to a reduced respiration or increased ventilation. So now I want to go a little bit more in the processes of the oxygen balance over these oxygen minimum zones. So as I said, oxygen minimum zones are located above major upwelling systems. So you have large inputs of nutrients, which is fueling a very strong and intense productivity. You have very high rates of photosynthesis. This organic material sinks, and this leads to very strong respiration at very high levels of uptake of oxygen at subsurface. And this respiration is partly balanced by the ventilation of oxygen, but these oxygen minimum zones are also located over shadow zones, which are zones where there's actually very weak ventilation by the mean advection. And so most of the ventilation actually in these areas is achieved by mixing processes, and some of them due to eddies, as has been shown by Laure Esplandy, for instance. And so at equilibrium, there is a balance between this level of respiration and this level of ventilation. And the balance is shown here by the fact that these two circles have the same size. But the question is, what will happen under some change? Will the OMZ expand or shrink in response to climate change? So you can have a thought experiment and think of climate change as something that's going to increase stratification, for instance. So if you increase stratification, basically what you do is that you're going to reduce the exchanges between the surface of the ocean and the subsurface. So you're going to reduce the supply of nutrients, reduce photosynthesis, and therefore reduce respiration. But you're also going to reduce the ventilation of oxygen. So if this reduced respiration is actually equal to the reduced ventilation, then there should be actually no change in the volume of the OMZ. We'll just change equilibrium, but with no change. So it's very subtle, and the response will depend. So there are two cases: either the reduced respiration will exceed the reduced ventilation, in which case you will have a contraction of the OMZ, or the reduced ventilation will exceed the reduced respiration, and in that case you will have an expansion of the OMZ. So now, moving from this thought experiment to numerical experiments, I want to highlight how this happens in one specific OMZ, which is the oxygen minimum zone of the Arabian Sea. It's not the biggest oxygen minimum zone in expansion, but it's really the world's thickest. And it's an OMZ which has a very intense core, where which is very suboxic, and where denitrification occurs. So I'm going to sum up some studies that I've done in collaboration with a group of people working in the Center for Prototype Climate Modeling in NYU Abu Dhabi, Zouer Lashkar, Parvati, and Al-Azhar, and showing a set of different model experiments where we've been exploring the response of this specific OMZ to changes in local forcings. And for that, Zouer has set up a high-resolution regional model of the Arabian Sea OMZ. Here I'm showing some results from this model compared to observations, which shows that the model fairly well reproduces the core of the OMZ and the total volume of the OMZ compared to observations. Before moving to the response to local forcings, I want to stress the importance of model resolution for resolving this specific OMZ and also others OMZ. Here I'm showing a set of 4 model experiments. It's the same model. The only difference is the grid resolution from 1/3 of a degree to 1/24 of a degree. So moving from left to right, we have finer grid resolution, which allows the resolution of more eddies and therefore of more eddy mixing. And you clearly see that the impact is the shrinking of the core of the OMZ due to this extra eddy mixing, and it's really this highest resolution model which corresponds the most to what is observed. Now I'm going to go one by one to different factors that may be affecting the change of this OMZ. The first one is the warming of the waters, and particularly of the Arabian Gulf waters. Here is a map of the sea surface temperature trend from 1982 to 2010 over the region. And you see that the largest change in sea surface temperature is in this Arabian Gulf region. And this is important because the waters coming from the Gulf are actually coming inside the Arabian Sea and are providing a ventilation pathway within the Arabian Sea. So with the warming of these waters, there's actually a very strong reduction of this ventilation. And Zwer has looked in this paper at how that was affecting the OMZ. So on the left here, I'm showing a vertical section across the OMZ here in his control simulation, showing the OMZ core in purple and the OMZ in pink. And on the right panel, it's a Gulf warming simulation where there's a shutdown of the Arabian Gulf ventilation. And you see, by comparing the two, you can clearly see the expansion, the intensification of the core of the OMZ between 100 and 300 meters here in response to the shutdown of this ventilation. A second example I want to show is the response to Munson wind intensification. So there's some belief that in the future the Munson winds are going to intensify. When we have stronger winds, what is expected is to have a stronger upwelling and therefore stronger primary production. Therefore, we expect a larger respiration and a larger ventilation also due to the stronger winds. Zwer conducted different experiments with different winds, strong winds in red and weak winds in blue, and this shows vertical sections of the volume of the OMZ for the two experiments. And what you see is that there are different responses to stronger winds in the upper OMZ here and in the lower OMZ there. So by looking at the trends of respiration and ventilation, we were able to understand why these were not behaving in the same way. In the lower OMZ, where there's an intensification of the OMZ, you see that the increase in respiration, which is shown here by this green curve, which shows over time the change in the respiration when the wind is increased, occurs much faster than the increase in ventilation. And this explains the intensification of the OMZ. In contrast, in the upper OMZ, it's the opposite. The increased ventilation here in blue occurs faster than the increased respiration, leading actually to a shrinking of the OMZ and of the OMZ core. Finally, we looked at all of the different factors together in a hindcast simulation covering the period 1982 to 2010. And this plot here shows the time evolution from 1992 to 2010 of the suboxic volume anomaly in this simulation. And the first thing that you notice is the strong interannual fluctuations, which are really very strong and make it difficult to detect a long-term trend. Nevertheless, there is a trend for recent expansion of this core of the OMZ, of this suboxic volume. And this expansion is associated with a deoxygenation signal in the north of the Arabian Sea. So this is the total deoxygenation trend in the control simulation. What Zwer has done is, in order to understand what was driving this trend, he conducted a series of simulations where he changed the forcing. So for instance, in this one, he didn't consider changes in the wind. He kept climatological winds. In the second one, he didn't consider any warming at the surface. And finally, in the third one, he didn't consider warming except in the Gulf. And by comparing the changes, the deoxygenation trends in all of these simulations, he concluded that the wind and Gulf warming together are not sufficient to explain the simulated OMZ expansion over this period of time. And in fact, the main factor, according to this study, is really due to a reduced ventilation associated with increased stratification. This increased stratification is due to the warming of surface waters, locally. Now the question is this trend going to continue in the future or not? And for that, what we have in hands is these CEMIP 5 large-scale global coupled model simulations, which are coarse resolution. Here I'm showing the oxygen minimum zones across this transect in observations and in 3 of these different models, the NPI model, the GFDL model, and the IPSL model. And what this comparison shows is that actually none of this CEMIP model reproduces the oxygen minimum zone correctly. They either strongly overestimated it, like the MPI model, or strongly underestimated, like in the IPSL model. So what Parvati has done is that instead of looking, in addition, I would say, in addition to looking at the trends in this model, she has used the anomaly of the forcing in this model to force regional downscale experiments. I'm going to show you this now. So that's the projected response of the change in the volume fraction of oxic waters over the 21st century. First, for these CEMIP simulations. So on the top, it means there's an expansion of the OMZ, and then on the bottom, you have a shrinking of the OMZ. You see the changes are fairly weak for the 3 models. The MPI projects a shrinking, the GFDL projects a very small expansion, and the IPSL projects no change. Now, if we look at the results of Parvati, in her downscaled experiments, and this is really a work in progress. So she's still trying to understand what's going on. But if you look at the results of our downscaled experiments, you see actually a shrinking of the OMZ in all of the experiments. So she's run experiments using these 3 different forcings for the 3 colors and 3 different model resolution from 1/3 to 1/12 of a degree. All of the experiments project a shrinking, which goes in the opposite direction as the trend that we saw for the recent period of time, where there was an expansion of the OMZ. So this suggests that it might be due to a stronger contribution of reduced productivity in the future compared to the present period where productivity, there's no very clear trend in productivity and respiration. Another question is the sensitivity to model respiration, to model resolution, sorry. And interestingly, it doesn't go in the same direction for the different forcings. And these are also things that Parvati is trying to understand at the moment. So I'm just going maybe to give some key messages. Global deoxygenation is something that we've been able to observe and project with a high degree of confidence. And this is because it strongly depends on the increase of sea surface temperature, of the temperature of the ocean. And it's fairly easy to quantify. On the other hand, it's not the case for the OMZ evolution, which depends on the subtle balance between changes in respiration and changes in ventilation, and for which we still have low confidence in the evolution of oxygen minimum zone. And understanding the future variations requires really precise estimates of the changes in respiration and ventilation, of their time variations, of their depth distributions. So you can have different responses over time at the surface, subsurface, etc. And maybe the main message is that global deoxygenation, given that, is not systematically associated with the expansion of the OMZ. One study that I really like, which shows this very clearly, is this study by Ball et al., which used a global Earth system model where they have only changed one parameter, and they conducted 6 simulations by only changing the mixing coefficient. And all of the curves here show the deoxygenation for all of these simulations. So all of the simulations show deoxygenation. They're all on one side. But now if you look at the change in hypoxic volume, you see that some of their simulations suggest there is an expansion of the OMZ, and some others project there's a shrinking of the OMZ. So really, it's two different things, which is the global deoxygenation and the change of oxygen minimum zones, and we really need to focus on the very small details of ventilation and respiration to understand what's happening in these very sensitive oxygen minimum zones. Thank you very much, and I'm open for questions. And let me stop sharing my slides. Can I do that? Stop sharing, and I'm here to answer your questions. And I know Zuer and Parvati and Damien are also here listening to us, and so they're also be able to answer to more specific questions about their work. I also like to stress that Parvati and Zuer, their contract is going to end within 6 months, and so they will be open to any opportunity that you can offer them. Thank you very much.
Wonderful. Thank you so much, Marina. It looks like we have a few questions already queued up. So Suresh is asking 2 questions.
Why is my video stopped? Yeah.
There we go. We can go. Gulf waters are warmer and hence lesser O2 saturation. How does it contribute to oxygenation of the Arabian Sea OMZ? Or did I miss something?
Sorry. Do that. Let me read the question. Gulf waters are warmer and hence lesser oxygen saturation. How does it contribute to oxygen, or did I miss something? Hi, Suresh. So Gulf waters, when they actually hold oxygen. So when they get warmer, they don't penetrate as well. They don't ventilate the Arabian Sea as much as when they're colder. So there's a lack of supply of oxygen when they get warmer.
And it looks like we've got another question from Greg. Marina, would you prefer to read them? Is that easier for you, or do you want me to?
Yes, I think it's easier if I read them. Thank you.
Okay.
Right. With respect to all models, including the one for the St. Lawrence, respiration in the water column are just included. But what about benthic respiration, which is a long-term factor given the amount of carbon in this reservoir? So I can only answer for our model, but in our model, there's no benthic respiration that I know of, and Zuer will correct me if I'm wrong. But the OMZ is well above the bottom of the ocean anyway. So I'm not sure this would have an impact. Not sure if I answered well. Maybe Mathilde, you want to answer that question for the Saint Laurent?
Well, I can say just for the St. Clarence, so I showed is all based on observation, so it wouldn't have like any modeling contribution. But what we do is we look at the total loss of oxygen, so it would include both benthic and the pelagic.
So then another question. In one of the initial slides, you showed increasing resolution shows decrease in OMZ, but the last slide from Parvati, yes, the last slide from Parvati's work with increasing resolution does not seem to follow this. Yes, because that's fair. Yes, it's a very good question, Suresh. There's a difference between the first slide and the Parvati's work, which is in the first case, we were looking at the OMZ with no climate change forcing in addition. And Parvati looked not at the change of the volume of the OMZ, but the response of this volume of the OMZ to a climate change scenario. And what Parvati has shown is that it can go in both directions with resolution for different forcings, and we still don't know exactly why. So she's investigating this at the moment. Yes, right, not on the margin and horizontal transport. But this is true, but I guess at least in the case of the Arabian Sea OMZ, I mean, it's really a huge body of water, which is not only on the margin. I mean, there's a lot of it is not on the margin, and so I'm not sure how much this benthic respiration would account for in the total respiration budget. Okay, so that's in French. Then, to what extent are the changes in respiration driven by changes in primary production in the water column in these models? I'm not sure I understand the question very well, but the changes in respiration are entirely driven by changes in primary production in these models. It's really the change in primary production that drives the change in respiration. They're very strongly connected. That's how I can answer to that question. Maybe you wanted something more precise than that. I'm not sure.
Heather, do you have any questions that have come directly to you?
I don't see any in the chat, but another one just came in the Q&A. It looks like two more. Mesoscale eddies.
Yes, so we've looked at the role of mixing by increasing the model resolution. We went as far as 1/24th of a degree, which is mesoscale resolving and maybe sub-mesoscale permitting. So going from 1/12th to 1/24th, we have a little bit of sub-mesoscale. We'd have to go a little bit further, I guess, in resolution to really quantify more precisely the role of sub-mesoscale features. But there is indeed a very big role of mixing by mesoscale eddies, and this is true for all oxygen minimum zones, and this is really due to the fact that they are really located over regions where there's very low ventilation, and because oxygen is consuming, respiration is strong, and is consuming this, is creating very strong gradients in oxygen, and so that leaves, you know, a very strong role for mixing to work on these very strong gradients, and particularly mixing by eddies.
Okay, I think we have time for one more final question from Bastien.
Regarding the change in respiration, can the models account for changes in community and downstream effects on remineralization depths and therefore depths of the core? Yes, that's something that we have actually explored. It's the work by Al. I didn't mention this work, but he looked at particularly at this question, at the effect of the remineralization depths. He compared the situation in the Arabian Sea to the situation in the Bay of Bengal, where the remineralization depths are fairly different, and it had some very important impacts on the OMZ. Yes, it is an important factor. Okay, thanks, Greg. We'll keep that in mind. Thank you. It's something that we might want to tell. Thanks.
Thank you, everybody, for joining us today. I want to thank our speakers for just phenomenal talks. We really enjoyed them. And I want to thank Emily for joining us as our guest host for this web conference or webinar. And I've already dropped the link for our informal chat starting in about 10 minutes with our speakers. That's plenty of time to grab a coffee and some slippers and come on back for some informal chitter chatter with our speakers.